Welcome, everyone. I am Natalie Kaiser. I'm going to be the host for today's NAPB webinar, and we are thrilled today that Dr. Patricio Munoz is joining us, and he is going to talk to us today about blueberry breeding. Dr. Munoz is an associate professor of horticulture sciences at the University of Florida, where he focuses on the development of high-yielding, high-quality, and better disease and pest resistance in southern high bush blueberry cultivars. In his program, he explores the genetic variability of commercially important blueberry traits using traditional breeding tools and quantitative genetics with the aim of increasing genetic gain in selection traits and optimizing the generation of new cultivars. His interests span discovering, developing, and applying molecular marker information to advance plant breeding through marker-assisted selection and genome-wide selection. And with that, I'll turn it over to you, Patricio, and we're excited to hear what you have to share with us today.
Thank you, Natalie, for the introduction. I hope everybody can hear me well there. So I'm very honored to have the invitation to speak to all of you. You know, finally, we were able to match the times and make this happen. So today I'm going to be talking to you and then from the point of view of a breeder. And I'm going to be telling you, well, how our breeding program works, but more importantly, what methods and technologies and protocols we are developing to do what we do better. Right? So let's start. So probably the first thing that is important to be aware, why do we do what we do, right? Is because blueberries keep increasing in consumption. And so consumers are demanding for more blueberries, and then there is more production of blueberries, which means that there is more demand for better blueberry varieties. In my team, we kind of have two sides. One of them is the breeding program that, you know, has a whole operational team that works on that. And then we have the research program. So in the breeding side, you know, we do development of cultivars. We usually say we local focus on global impact. But also in the research side, we do research to implement any, in reality, advanced methodology or any methodology that can make our program or breeding program to do better, faster, more accurate, more efficient, right? And then probably I'm going to talk about both of them. So let's start with the breeding part. So in breeding, the University of Florida has a huge, it had a huge role in the development of blueberries, but knowing the standard or traditional northern high bush blueberry, and that's why Natalie was referring at the beginning that I work with southern high bush. Originally, blueberries were domesticated in the north, in New Jersey, right? And those blueberries require a high chill or deep winters for the people that is not familiar with chill requirement in order to produce, right? So Dr. Sharp, Wade Sherman, and other associates, they started working in 1950 to hybridize these northern ones with native ones from the south, of the southeast, and specifically from Florida. And you know, the first variety 26 years later came to light, and that was Sharp Blue. Now, what I always say is that Dr. Sharp wanted to bring blueberries to Florida, but in reality, he didn't get to realize that with this new development or with the products of this hybridization, he developed a whole new varietal type, right? That we call collectively southern high bush, or we also call it tropical blueberries, or we call it no-chill blueberries, and many different names. And then since that moment, you know, the program, and after, you know, I'm the fourth breeder after Dr. Sharp, the program has released more than 68 varieties directly, and then more than 80, and probably we're getting close to 100 with the different collaborations that we have globally. Not just the varieties, but also the program has focused in innovation and creating, for example, discovering varieties that they were aromatic in the past, or discovering varieties or developing varieties that were crunchy that nowadays are you can find more and more in the supermarkets. And also, for example, varieties that they have different colors. Now, for my fellow breeders and students or younger breeders, I always say, plant breeding works. So it's not that we need to fix plant breeding. We're just trying to do things better. So for example, this is a plot where we have the year of the release of the cultivar, and then we have the firmness of the fruit, or the firmness of the blueberries in this case. And then you have the different colors for the different varieties. And we can see here that through time, and then with the work of breeders, we have been able to increase significantly the firmness of the fruit because nobody wants a mushy blueberry, right? Now, regarding our program and what we do is we license these cultivars globally. And that's where what I usually say, the global impact of our program. And in that way, we also have partners, global partners that work with us or in different parts of the world. And that's the way that we get our blueberries, to the consumers. And then I'm going to go very quickly through a few examples that you might hear if you are in the area of blueberries, of course. So this is some of the newer varieties you can say from our program. For example, Colossus, with a large blueberry, Sentinel, very high yielding and very precocious variety. By the way, the Southern High Bush, or the varieties that we work with, the program start working, for 30 years in actually to make them precocious and more and more precocious. And nowadays, our blueberries, they are able to yield in year 1 after planting. So which is a huge, is hugely important for the producers because they are able to start getting, some returns right away in year 1. Or Kikris, that was one of the crunchiest varieties that is out there in the market. For example, Albus, which is very liked by consumers. And this is, for example, pink variety that I was mentioning before, or pink or yellowish or white is an albino, variety. Some of our new varieties, and obviously the capacity to yield here. This is a true picture that I got actually a few seasons ago. And for example, also another of the premium varieties that we recently released that is very high yield and basically is the one that so far is our newest variety and the one that has been checking all the boxes that as breeder we are looking for. So the important thing is that consumers are willing to pay for quality. And it's the reason that this is a survey, for example, as part of the back cap, our one cap USDA CRI grant that we have with many of my colleagues across the country. And this is, sensory panels where we have different varieties in the different dots, and then we have the consumer liking and the willingness to pay. And then in here, we can see that the people is willing to pay more whenever they increase their liking or the quality of the fruit. And you can see, for example, one of our varieties over there. Now, if that's the case, that means that in our programs, we need to understand a lot more about the consumer overall liking, what it is, what are the characteristics that influence, the consumer. And then obviously we're going to have, the appearance that we have a bias as soon as we see something, right? And then we have the texture and then ultimately the flavor. And the flavor is a lot more complex, as you might imagine, because we have these 5 different things that you can test in your tongue, but you have hundreds maybe of different volatiles that they also fit, are sensed in the base of your brain. And all this combined actually, is what it gives you the overall like. So we work a lot in this area to try to understand this very well, match it and then link it to our biochemistry of our blueberries or appearance, for example, too. And then try to use that as a guide to what characteristics we should be pushing. So our program is very data driven in that regard. So in one sense, we have the consumer that is going to be looking on, is going to be making selection of what he eats based in obviously, people usually says budget. Yeah, budget obviously is going to be the first more important the most important characteristic that consumers are going to be using to decide what to buy, right? But after budget, you know, you're going to have these different things that consumers think. So somehow we have methods, different methodologies and tools to actually to try to develop something that consumers are going to appreciate. So that's more or less regarding the breeding part of my program. But we do a lot of research in the program, and the research that we do focuses into do breeding better. So my research program is divided in 4 areas, and here we have the leaders of the different areas. So we have Felipe Ferrao, Juliana Benevenuto, Felix Enciso, and Bruno Leme. And so we're going to be touching a little bit on each of these areas and what do we do in these areas and how we are being able to make some progress in some things that are actually already integrated in the breeding program. So let's start with the statistical genetics. So this is probably the oldest branch that we have in the program, led, like I mentioned before, by Felipe Ferrao. And so this program focuses mainly in 2 areas. One of them is kind of gene discovery and then the understanding of the mechanism, you know, and everything that it has to do with quantity genetics and quantity genomics and obviously genomic selection, right? So then, but obviously we focus in a few traits that they might be more important to our consumers and also some others that might be more important for our producers. And some of the people here actually has graduated already, is working in some in companies already. And in the other side, we have the methods and techniques that actually we have worked to try to integrate it in the breeding program. And not surprisingly, a lot of these things we already integrated in the breeding program. Like I said before, it's the oldest branch in the lab. For example, this gives you an idea of all the QTLs that we have discovered through the years. And we have here QTLs for fruit quality, you know, for fruit set, for diseases, for volatiles specifically, you know, so to influence flavor. And basically, a lot of them are being integrated into our either marker resistant selection or genomic selection panels that we are using every year. Regarding genomic selection, I know there is a lot here, but it's a pretty good way, and you know, I really appreciate Felipe to do this. It's a pretty good way to summarize everything that we have done regarding genomic selection. So here you have all the papers in this area here where you can find all the information. On the top, you have the software that we have developed. We are not a lab that focuses in developing software. I mean, let me just make that very clear. So we do this only when there is a need that we cannot solve with a software that is out there. And for example, at the very beginning, whenever we developed AGH matrix with Rodriguez, because there was no software actually to do the A matrix using pedigree, right? Well, in reality, there was a software, but you need to pay a lot of money. And then, so we developed one that was for free, right? So still a lot of people use this. And in the case of UpDog, I mean, this is the case of Felipe and in their collaborators that developed this is the one that we actually use to call allele dosage. And then through other collaboration, of course, you know, with a great collaborator, Jeff Endelman or Marcio Resende, we have been doing other softwares that we are currently using in the breeding program. Now, you know, pre-genomics, I mean, we still have a very large pedigree that it took us around 4 to 5 years to curate and put everything together. And this work started before I arrived actually in the program. Now, whenever we start using genomic selection, I mean, something very important, and I think some of the issues or some of the challenges that we deal as a breeder is what to cross with what, right? So then obviously then we start using optimizers right away to try to, because these are very large populations that we have in the field, right? So genomic selection, we start probably, I would say maybe 2018, 19. And then we were at the beginning limping because, I mean, our populations or training populations were not the best. But I mean, this is my advice. I mean, you need to start, the best moment to start is now. And then it's not going to be perfect, but it gets a lot better with time. And then obviously with more data. So then currently, you know, genomic selection is fully operational in our program. It runs, you know, very nicely. And now we always focus in doing small tweakings or small changes that actually that make it even better. So this is more or less how the program runs. So we have all the way from the crossing, and this is not going to be very different from other breeding programs, right? Maybe the difference is, for example, that we used to do genomic selection here, tasting panel, biochemistry, and then a lot of these different tools and methodologies that we implemented. And then the idea was to do rapid cycling with these things. Nowadays, probably the starting actually this year, and in a few weeks, basically we're moving genomic selection from this stage to this stage. And that's probably going to be it's going to have a huge impact. Why? Because we have studied so much to try to make this decision. And at the same time, we see that we have simulated all this already. So we know that the impact that we expect to have in our program. So the generation, the breeding, generation or the breeding cycle is a very important, is fundamental for breeders. The faster that you can do that cycle, the best. The more the genes that you can accumulate, the more that you can accumulate the frequency of the genes that you are after, right? So at the beginning, when I arrived, the breeding cycle in the past maybe was 6 to 8 years, because obviously, like in many perennial crops, you will want to make sure that, these parents, they have the right characteristics. We were able to short that to 3 years, to 4 years, sorry, and then to 3 years, and now we're trying to do it in 2 years, and now we have other methodologies to keep decreasing that breeding cycle that I'm going to show later. So it's so for perennial crops, or for crops, from long breeding cycles, is an essential part. I mean, it will give you a lot more gain that actually, implementing other methodologies, in my opinion. And that's why, since the beginning, we have focused in doing that. Some other areas of work of these areas, for example, metabolomic selection that we use operationally in the program. So this is the flavor wheel that we built in the past. So these are all the volatiles, so most of the volatiles, and this is basically how humans sense them, right? And then we have proven, with some studies in the past, that actually these volatiles, they are more important than sugar and acids in determining the overall liking of consumers. And that's the main reason that we put a lot of effort in this area. The other things is, like I mentioned, we are very data-driven and then consumer-oriented in our data. So, for example, it's not that you just keep increasing the traits, but actually, there is some thresholds where the consumers it really impacts the consumer. So we use these new methodologies actually to decide until what point do we need to push a trait. And in that moment, I mean, you don't need to keep pushing. The marginal effects are not enough. And for example, at the same time, a classification compared with a prediction or a correlation is going to be a lot easier to do. So we can increase the accuracy. So there is other ways that we can keep playing with this, and we are playing with those things. So the second group in my lab is the computational biology group, and that group is led by Dr. Juliana Benevenuto. And then I'll tell you, so this group focuses in these 5 main areas. So we have the wild species, building genomic resources, and getting variant discoveries, transcriptomic and target genes. So let's look at a few of the examples. In the context of wild species, blueberry is amazing. And here we put, there is 500 species listed in Vaccineus genomes, but actually this is kind of there was just a recent paper that this is kind of being redefined, because the borders between, the genus and other genus that are close by, they are so thin. And actually the taxonomy of blueberries has been so controversial. Just yesterday, we have a speaker here in Florida talking to us about this. The use of wild species has been essential since the beginning. And for example, we already have cultivars in our program that they have a big proportion of some of these varieties of snow chaser, or even kestrel, Vaccinia elliotii, that is very flavorful, very aromatic, or meadowlark that comes from almost like a tree, Vaccinia arboreum. and so on and so forth. So we probably in the near future, we're going to see this number to grow to 800, maybe 1,000 species listed under this, I don't know how even to call it properly. So under this group that eventually you could use to cross. So only 43 species have been used for this. So this is the work of Anissa Khalid in my lab that actually has been submitted and hopefully is going to come out soon. But only 43 species out of the hundreds that are out there. You know, she did a, you know, complete review of the literature and even talked with some of our colleagues to try to find more about this. And it's been fascinating to see all the potential that exists out there. So more about that pretty soon when the paper gets published. Now, use a couple of examples of how we use wild species. For example, this is Vaccinia elliottii, and that's the wild species. So we standardly in our program, we double the genome, and this is a tetraploid elliottii with the genome double. And then you can see the F1, the F2, and the backcross 1. So already by the backcross 1, we already are able to recover most of the characteristics of southern high bush that we have here as a comparison. And this is a paper, you know, of one of my master's students that also now works in a company. So another example, and the one that we have been working a lot recently, is Vaccinia staminium. Why? Because it has a lot of good characteristics, but one that is very interesting, that it has maybe a different composition of anthocyanins in the flesh, and also is able to produce anthocyanins in the flesh. You know, traditional blueberries are going to be whitish or greenish, like this other Vaccinia staminium variant that you see here. Now, in this area also, they work a lot in genomic resources, and this is not to try to say that we did this. Actually, this is our colleagues, and that's what they provide us with different, you know, different genomes over time. And actually, you know, working with as part of the backup again, the USDA CRI project. So we work together to release this pangenome. But the question has been, how do we use that? And then we still needed, you know, a very good quality southern high bush that is now almost ready to come out. But in reality, now we have 4 or 5 in our program. And still the question, and I'm going to be showing you in a second, you know, how to use that pangenome in a way that is really usable for breeders, you know. I mean, and again, this is obviously from my opinion. Now, what are we doing with these genomic resources? You know, as soon as Patrick, you know, Edgard in Michigan State University, you know, told us he had the genome for Draper, the improved version of Draper genome. So we got access to it, and then we actually wrote this very nice and short article that I would recommend, you know, colleagues that they have less genomic resources in your crops to read. So basically, we went from, you know, a draft to a full genome, and it has huge implications in, you know, the way that we interpret our results, the number of pigs that we found, or actually the genetic architecture that we thought, and actually the ones that in reality is, right? So we also see an increase in or similar accuracy when we drop a lot of the molecular markers or the probes that they were not needed in reality for us to do genomic selection. So and that was also part of one of the optimization moments of the program. So how do we use the pangenomes? I mean, I think that we are working a lot in this area. And then again, I have one more slide on that. But in reality, you know, something that is going to be very relevant, especially with the topics nowadays, you know, with CRISPR and all these different methodologies of gene editing, is actually to build a good gene atlas to know how the expression of every tissue or the genes, how they express differently in every tissue. And that all this information, basically, we're probably going to be using it in the near future, actually, to make decisions on what we work on, right? Some example of the target genes, for example, we work a lot with linalool. And I know I've been discussing this for a while now, but linalool is a very nice example of a pig and replicable, I mean, a pig that we found. And then, you know, the genes and the validation of these genes and how this is very impactful positively to the aroma or to the flavor or the overall liking of consumers. But also we can go backwards, and actually here's where the pangenome comes in. It's like whenever we find a gene that actually is interested in other crops. What happened in blueberries? We ask the question all the time, do we have this gene in blueberries? And in the past, we have a very probably process like, it can be very fast to find out if it's in blueberries, but then it's on what, So nowadays we have created, a database and an interface that actually in our program that uses all the assembled genomes that we have, plus all the pan-genomes, plus all the whole genome sequencing, plus all the molecular markers that we have, and it uses everything in one single tool that whenever we put it here, any gene, it gives us a whole picture of what individuals are carrying what are the variants, does it exist, where is it located, and all these things that actually we are using to make decisions to what do we focus on and whether this is worth pursuing or not. So regarding pursuing or not, the genes or specific areas in the genome. So then it comes the cellular and molecular biology part of my team that is led by Dr. Felix Enciso. And in this group, it has different task, maybe I should mention that. So we have all what is marker assisted selection. They do gene validation. They do the pipeline for gene editing, and so on. So for all the things that a molecular cellular molecular biology lab does, and obviously all the tissue culture as well. So regarding the gene editing pipeline, it's going to be very essential for us to have pretty good regeneration protocols. And then just to give you an example here, so the way that we started, this is the whole variability of blueberries, or actually our breeding population. So we selected individuals and we were able to study them to try to find out what are the protocols that are going to be more readily available or widely applied to or germplasm. Right. So then obviously we were able to find, pretty good protocols. So this was the first step. And then in the second step, we say, okay, now let's, push even more. And nowadays we have a study where we are doing 180 different genotypes. We are being able to regenerate most of them successfully. And obviously all the tissue culture that is prior to this has been established in the program for a lot longer. So the second step in the gene editing pipeline, if you are using traditional gene editing, I don't know if that even means something, traditional gene editing, but if you're using transformation as a way to get there. So basically, we have tested so far 3 different protocols, and with the expertise that we have in the lab, we're being able to make 2 of them work. And here is one of them where you can see the control and the transform, and then a pretty beautiful picture there of a transformed plant with a reported gene. Now, we are in the path to optimizing this protocol. We always hear in the past, transformation protocols of 1 to 3%. So we are very happy to see that we are being able to do up to 8 or 9% with our protocol. And then we're still in path to optimizing. So this should get a lot better. Second different protocol that we are also testing in this area. So we're able to see, pretty good signal. And in this case, again, like I said, we're testing 3. We have tested 3, 2 of them work. And in this case, we're able to get 5%. So this made me very happy because in reality, we've been working for this for quite a while. And it's been very difficult, honestly. Blueberry is nothing is easy in blueberry, honestly. And then but these numbers are very encouraging. So marker assisted selection, again, as one project as part of the backup and this big USDA ACRI across the whole country. So the idea was to actually develop a marker assisted selection, but not just to develop the assays, develop the protocols, and also, study the cost of the different things. So then actually, last year, we applied this new methodologies in the program, and then we were able to do, selection for some of the traits that we are using in the program. So that obviously this area also is under the cellular and molecular biology. Excuse me. Finally, the last but not least area, and actually the newest area on our lab is the fruit phenomics, we call it. That is the one that is in charge of automation, equipment, machinery, and all that. So in this area, for example, something that is very, very important for us and very problematic. I know they're breeders of blueberries or small fruit will understand me. And especially if in your crop you have the same issue than us, and blueberries, they need to be harvested every single week. We don't have a single harvest. So we harvest every single week, and that could last up to, in some cases, up to 12 weeks. So it's a huge issue. So we have developed methodologies nowadays to do berry counting and yield estimation, and then that's already being used operationally in the breeding program. Now, that's not fully automated, and then this year, actually, we are about to deploy them. I mean, actually, probably they're going to be testing next week the deployment of this automation that we are working in, and then we decide to do rover instead of drones because of it has a few. We have a few things and also political issues that to use drones in the state. But this is the milestone for this year, so it's going to be exciting. So this team in 1 year was able to give us a tool that we have used already operationally, and this is the second step in that direction. The other thing that we are using is we, like I mentioned before, crunchiness or texture of the berry is something very important. And then we have been developing the protocols to do classification of, using what you see here. This is a texture analyzing, crushing the fruit, and then we have microphones that are, collecting that data, and then with that, actually, we are able to put it in a classifier, and then we're able to classify, what is the different textures of this berry. Now, obviously, this is, a lot of work, right? I'm very slow, but the idea is to use the data for us to develop prediction models and obviously to try to find out the genes that are, related for the traits that really matter to consumers. Another thing that this team does is to develop tools for us that we really need. And for example, these are the schematics of the different modules that this carousel has that we are developing to measure the traits that we want to measure and we need to measure, and then they're too slow to measure otherwise. And then obviously, he is using all the new technology and all the other things that we have in software. So we are using image analysis, and now potentially we can do it in one single stop. I think the other thing, and something very important, and now I tied back with what I said before, in the case of perennial crops, essential to decrease the breeding cycle. So far, we have decreased the breeding cycle using all the tools, molecular tools, genomic tools, and everything, a ton of logistic to decrease the breeding cycle. Management, you have that, we have all that. So what is the next frontier for us? And this is something that we have been working for a while. And now the prototypes we have developed it, and actually we are in the process of developing this whole set. But this is the schematic of how it's going to look like. It's going to be, or basically, or recipe development, chambers. And then with these ones, we are able to control temperature, humidity, and obviously all the aspects of light. And then whenever this is ready, we already have acquired a couple of these containers where all these recipes are going to be implemented. Why? Because we need to decrease the breeding cycle even more, And with that, probably will be the shortest that we're going to be able to make it happen. And it's going to have huge implications, not just, in decreasing the breeding cycle, but in integration of genes and using wild species, gene editing, and everything that we are doing. So I'm sorry that I talked very fast, and there is a lot of different things here. But blueberry, it is what it is because of the health components, this healthy halo that it has. And then it's being classified as superfruit. We keep mentioned this at the end of my at the end of my talks always, stay healthy, keep eating blueberries because they are good for you. And I think with that, I'll be happy to entertain questions and comments.
Thank you very much for sharing all the diverse facets of your program. We have a couple of questions that were asked ahead of time and one in the chat. For all the participants here, if you have additional questions, please drop them in the chat and we have plenty of time to address them. One was a question about high throughput assays for effectively selecting for anthocyanin content. You showed closer to the end some of the high throughput tools. Does that enable selection for anthocyanin?
So anthocyanins, there is an aspect of anthocyanin that's very environmentally controlled, right? So we are working with anthocyanin heavily. So we know that we have genotypes that they are more stable through the, different environments and also through the post-harvest period, right? And then the idea is that we are trying to develop more of those individuals and not the ones that, they vary everywhere, right? Now, for assays, we do have an assay that we are still validating. We still, I'm not going to say that we have it and it's done. We are validating still to make it more high throughput. And as you might imagine, color obviously is a big thing for anthocyanin, but it's not everything. Okay, so it's like basically I'm saying by just looking at color, you don't get everything so far, at least in what we do.
We have another question here, how long it takes from crossing through to the release of a new variety? What is that pipeline life cycle?
Used to be 12 to 15 years. The shortest that we have been able to cut that has been now 7 years, and the goal is to do it in 5 years.
A very aggressive shortening.
Yes.
We have, hello, great presentation. Any work related to produce blueberry hybrids by seeds? And also, what is the cost of cloning in blueberry?
So this is funny because just one of my team members the other day was mentioning about this, and then I said, no, let's hold this. We have already too many things going on. We have already too many, low-hanging fruit that we need to, or challenges that we need to address. No, we are not doing any work on that. And then I don't envision working on that anytime soon. The cost of a clone, the cost of a plant, again, in the nursery, I don't know, it could vary between $3 and $5, depending on, where you are buying that plant. Probably if it comes from tissue culture or micropropagation, or if it comes from softwood cuttings. Yeah.
There was a question also related to rootstock breeding. Is there any ongoing efforts for rootstock in blueberry?
So we have some colleagues in the university, Jeff Williamson, and I think now Gerardo Nunez, they are been working with one of the species I was showing you before is one of these wild species, Vaccinium arboreum, that basically is almost like a tree. So they've been doing some selection to select for monopodical, for just a single stem in order for rootstock to work. Currently, we do have a lot of variability within the germplasm. I think we could achieve that. Blueberries doesn't have the problem that other crops, for example, I don't want to make this big assumption right now, but we don't have, and hopefully give me wood, we don't have problems with nematodes, a huge thing. And then there is a huge variability, and then usually the pests and diseases are very variety specific. So then we are able to go around them by just keep working in what we do best, which is breeding and developing cultivars, right? Also, southern high bush is non monopodical. So that means that you have multiple canes that come from the bottom. So if that's the case, it will be problematic because if you can have one rootstock and if it comes from the same species, you're still going to get all these, shoots or stems coming and interfering with your work.
You mentioned in your talk breeding targets in the quality space and in size, which is a yield component. Are there any emerging breeding targets in the last several years, perhaps pests other than nematodes that you are worried about, or changes in consumer preference?
Well, consumers, we being a program that serves so many clients, right, is very important. And I will say for anybody that this is the case, it's very important to maintain a wide, germoplasm diversity, right? And this is something that we do very well. And so we see some changes, for example, in consumer trends, but we're able to respond to that, fortunately, because we maintain this, right? Now, pests and diseases are a huge issue, and then somehow we have been behind. We have to remember that blueberry is a tetraploid species, outcrossing, and then it has great things, for example, like the clonal propagation and also the being able to produce fruit in a 1-year-old plant, which is huge compared with other perennial fruit trees. But the genetics is very complex, right? So for us to, the protocol development has not been what we would like in other species. But we are catching up. So we do have, for example, already a 3 different students that have worked in different diseases. So we do have markers. We have nowadays genomic prediction models for these pests and diseases. But we have more. We have a lot more. I mean, it's Florida. Florida is a port of entrance of pests and diseases, right, Natalie? So, and what we don't have, we will have tomorrow. So it's a continuous challenge in that area. But it keeps us busy. So, yeah.
Pivoting over into a question about genomic selection, are there any traits that are still more reliable with phenotypic selection compared to genomic selection?
It depends. I will say what is your objective? I mean, in breeding, it's very easy to confuse things, right? So in breeding, we do 2 things. We do selection of parents and selection of products, right? So in selection of products, I'm not gonna use my tools to predict. I mean, this one I need to measure. I need to put the plants in the field and I need to validate, that. And then you're not gonna have anything more reliable than that. You need to have that. We need to have that data to show, because you are dealing with the pocket of producers and then to farmers that they rely for these things to work. So for product development, we put the products in the field and then we validate their performance. Now, given that it's southern high bush and no northern high bush, by the 3rd year, we already have reliable data to say that this works, right? In comparison, with our northern high bush counterpart, that by the 3rd year, they are getting probably the first crop or the first full crop. And that's the difference. And even in other locations that we have globally, that in 6 months since they plant, they can obtain a good yield already. So then it gives us a pretty good idea of the traits. Now, for selection of parents, I'm gonna choose genomics anytime because I can do it. I can rotate the program a lot faster. That's the way that I think. I don't know if this is the right answer. I mean, there is no right. This is the way I do things,
There's a question if you've seen any negative effects of selection for precociousness.
It gives you more work faster. No, not so far. And then we keep working in that trait. We keep pushing it. Because just seeing blueberries in year 1 is not enough. I wanted to have, full production by year 1. And so we keep pushing in that direction. And I believe that there is a huge potential on that. I mean, seeing some of our new varieties that are able to yield 10,000 pounds per acre, or 10,000 kilos per hectare, in year one, that's very encouraging. Sorry, compare it with the standard, that the standard was 1,000.
Okay.
So
There's a question, if any of your target environments have been impacted by labor shortages. You and I were touching on the impetus for mechanical harvest, but maybe you could give a brief history of mechanical harvest efforts in blueberry.
yeah, so this it's not just labor shortages, but also the increase in the cost of labor, right? So these two things, they particularly play a significant role in decreasing the profitability of the operations of our producers and farmers, right? And the breeding program, before I arrived, started working actually on developing varieties for mechanical harvest. By the time I joined, basically the work that it needs to be done, it was just evaluating some of these varieties. And just the year that I joined, I have the job to release one of these varieties that has been bred before actually I arrived. So in 2017, we released the first variety that can be fully, harvest with machines, mechanically harvest. That is called Optimus. And then, yes, the name is for Optimus Prime. If you have ever watched in the past, I mean, some people will relate to that, to the Transformers, because any variety that it has any machine harvest capability in our program is going to have a machine connection in the name, right? So since that moment, we have polish the methodologies that we use to determine whether a variety is going to be machine harvest or not. And we are putting a lot more focus on that. And then, 3 years ago, finally, the state harvests around 10% of all the blueberries for fresh consumption with machine. 2 years ago was 20%, and this past season was 33%. And then this keeps increasing. It's not just that the varieties are there, but also, I mean, I usually talk about the triangle. You need to have the right genetics, the right varieties. You need to have the right management, and that means even establishing the fields in certain way, the certain height, the beds, they need to have certain width. There needs to be a space at the end of the rows for those larger machines that they are larger than a tractor in some cases for able to turn. So just a lot of logistic in this. So that's management here, right? So genetic management. And the third one, obviously is the equipment that you are using, right? So the machine. So all this is being aligned, and we are being working as an industry more and more. But very importantly, now we have companies that provide the service of harvest with machines. And that was a game changer because, small producers that have, I don't know, 50 acres or even 100 acres, maybe it's not it's not a good investment for them to buy a machine that costs, $200,000 or $300,000, right? So then now, but if you have somebody that comes to you, it's like, say, hey, you know what, I harvest your berries, and then it's gonna cost you maybe a 30 or 40% of what it costs you to harvest by hand. It's a pretty good deal for both parties, right? So then that's what we have seen in the state. And this is going to keep getting better and better because these companies now provide us feedback of what varieties are harvesting well, and then we can adjust.
Have you discovered, developed, or are you in the process of developing any markers that are associated with machine harvestability?
I don't think we need them because we have proxy traits that they tell us very well whether a variety can be harvested or not. So, and again, because in that direction there is the line of product development, those ones we need to put it in the field. We need to run machines. We need to prove it. We need to validate it because again, it's not my pocket, it's the pocket of somebody else, and I need to be more careful.
Coming back to the precociousness question, do you foresee any impacts on plant health and plant longevity if production is started in year one at commercial standard levels?
I think that's a very valid question. And then it could happen. So far, it has not happened. The work is into build or into develop varieties that they are vigorous enough to rapidly build their root capacity, because we know that the roots are very important in perennial crops, where they're going to be storing what they need for the next season if it's in the evergreen production system, and I mean, sorry, in the deciduous system, and the evergreen probably will be a different story. And so in that way, you shouldn't impact. In the case of also, there is a huge difference between southern high bush and northern high bush, or the low chill and high chill blueberries, is that low chill blueberries, they don't last that long as they do the high chill blueberries. And it's obviously because we are pushing them more, but also because the weather conditions, they are more favorable. I mean, they sleep for longer in the north, while in the south they are working, they are awake most of the time, right? So the chronological age is different than, maybe the physiology. These things are awake all the time working, right? So then that has had an impact in the longevity of our planting. Now our producers are familiar with that length. Importantly, the varieties are being replaced not because the biological capacity of the variety, but they are being replaced because the consumers are demanding the replacement. And so we don't need varieties that last 50 years in reality, because the consumers are so dynamic and they change their preference. So we need actually to be able to respond to that. That's more important from my point of view.
What is the ballpark age or longevity for a southern high bush?
Southern high bush, I will say, people will argue with this, but I will say between 8 and 15 years, at least for Florida. But again, I mean, in some territories that are very aggressively growing outside the US, and then they can produce very, very rapidly, varieties, they are not lasting more than 7 years because the market is demanding more and better quality, and they keep pushing, so then it's like they just move very fast.
The question if there are any trade-offs or correlations between fruit size and quality traits. Does quality go down as you increase in size? And is larger fruit preferred for easy harvest or machine harvest?
In breeding, and probably all of my colleagues are gonna relate to when I say this. In the past, and then not very long past, very long time ago, whenever we have all these type of studies of the correlation among traits, usually was done between very few varieties that they were released in the market, right? And then there is reasons for that, because there is limitations in, production or fruit quality, labs, or physiology, machinery, equipment that is very slow, and all these things. And then obviously they couldn't do this with hundreds or thousands of varieties that we do in our breeding programs, right? So when we look at all these correlations that they exist in the literature, or they exist in the head of people because they have seen it, right? We are not able to validate any of those correlations. Usually they are a lot lower. And for example, fruit quality or size of the fruit with other characteristics, that usually, says larger fruit, softer fruit, or larger fruit, less sugars. I mean, we don't see that. We find everything in the breeding program. Now, if you look at a narrow group of individuals, probably you can find what you say here. Regarding machine harvest, for hand harvest, size is extremely important, because it increases the efficiency of harvest, right? And it's a component of yield too, right? So then it's very, very important that you have certain size. The efficiency of your crew harvesting these berries increases significantly when the berry increases the size, right? For the machine doesn't care, right? If it's knocking down a berry that is, 20 millimeters, or a berry that is 14 millimeters. Now, The market cares. The market cares. The market is telling us what size of berry they want to have. So that's more important to us that actually, the correlation that we find. But just to give you an idea, Colossus is a jumbo berry and it can be machine harvest. And in the other side, you have Optimus, which is fully machine harvest, also variety, but is a medium-sized berry. So both of them can be machine harvest.
Are you selecting for specific compounds that improve health, human health, for example, polyphenols?
Yeah, I mean, maybe regarding your question before, Natalie, of what are the traits or new traits, also, this is something that has been new, if you want to say, for the last 4 years, right? So it's something that we have been putting a lot of focus in the health components. So blueberry, it is, what I said before, because of the health components, right? But it doesn't mean that we need to make them even healthier, right? So then we are working on that heavily. And we are funded by a company to develop these blueberries that they are healthier. So there is different ways to get different ways to get healthier blueberries. Obviously anthocyanins or antioxidants are one of them, but the polyphenols is another one. Blueberry has many other components that they are good for human health, and we are investigating all, but one by one.
We have time for just one more question. Let's see if anyone is in the process of submitting. Do you think that markers that are being developed are applicable for northern high bush germplasm?
We actually tested that, and surprisingly they work. I mean, not for marker resistance selection, but our models of genomic prediction work, at least.
And then our very last, we'll end on a fun note. What extraordinarily novel trait would you like to add to blueberry if you could, if you think outside of biological constraints?
Outside? No, it's still inside biological, there is still a lot of things to do. So, I mean, this is something that I've been saying for a while. For me, what tomatoes have done is the model to follow, and actually what I'm following, and in my breeding program, and hopefully one day we will get there. That actually, tomatoes is like we do have different tomatoes for their function, the functionality of tomatoes. So you have tomatoes that people eat for breakfast, tomatoes that they are for flavor, tomatoes for sauce, tomatoes for this and for that. Well, blueberries has been blueberries, and then maybe we have 2 or 3 classes that you find in the supermarket. But I want to have blueberries of all the colors and all the sizes and all the shapes, and then that's hopefully the future. Functionality.
Like differentiation within
Yes.
blueberry. Well, thank you so much for sharing your time with us and your expertise. This has been really fun and insightful.
Very nice to be here, Natalia, and thank you for inviting me. Thanks everybody for attending.